what I want to do is tell you sort of something about topological data analysis, but really something about when and how you can use it. So I, as Jonathan said, I started and am still an algebraic topologist and a homotopy theorist. And so, it would do my heart good if this stuff worked really well to analyze real data. But, and I think that also, though, I come at this from a position of some skepticism about the degree to which this will actually work. And I want to communicate I think, some ideas about when this is really going to work and when it's not. So let me start. Yes, let me write some things now. So let me say I want to sort of start by explaining, at least at a high level, what TDA is, or is doing, and two, something about, maybe I can carry this with me, explain its use in scientific inference. And of course, the questions there are, what can this tell us? And when is it useful? Okay, and sort of some of what I want to outline here is a little bit about sort of an integration with statistical practice. And also, anything else? Yes, and also sort of something about conceptual limitations. So I want to give a view of how you would use this. There are lots of talks one could give about, sort of a basic introduction to combinatorial algebraic topology, which is what this is. And I'll do a little bit about that. But people have been thinking a bunch with varying degrees of success about how to do statistics in this context, and about what you can and can't ask, and what you can and can't hope to get for real, not just in some asymptotic setting from this stuff. And I want to say something about that. Okay, so let me start by starting from the beginning.
Yeah. You can start on the left and all the way straight.
Okay.
Just right high.
Yeah, okay. So I think the starting point for all of this is that, the general religion here is that data has some kind of shape, often, and some kind of qualitative, global invariants of that shape are useful. And this is the dogma, and this dogma is, I think, unobjectionable. In the sense that we know, what tells you that something like this is true. The basic thing that people do to analyze data they don't understand is they cluster it, right? You do a little bit of dimensionality reduction and you cluster. So it's sort of a standard thing to do. Standard pipeline is you do some kind of dimensionality reduction. And then you cluster. And so, you have your data, which you've projected into R2, and you cluster it, and you hope that these clusters line up with something else that you know or would like to know about the data. So the success of this, the sort of ubiquity and success of this tells you that, it probably really is the case that the shape information is useful. And now the point of departure for topological data analysis is that, this idea of qualitative global invariance is what algebraic topology is about. So, what this looks like, sort of, to a topologist, this looks like finding what are called path-connected components. Right? If you have a space, so now I'm going to shift for a second and think about continuous objects, so not discrete sets of points, but continuous spaces. And if you have a space, it falls, you can count how many pieces it falls into. Okay, so if I was better at this, I could draw exactly the same little bean pictures. But if I have this space, well, a qualitative global invariant of the space is the number 3. Right? And this is an algebraic invariant that's counting these components. And here, path connected means, for any 2 points, I can draw a path that stays within the thing. Okay. Now, it turns out, and I'll explain this a little bit in a second, that it's better not to think of this as 3, but to think of this as a 3-dimensional vector space. So maybe R3. Okay? So we want to think about that an invariant associated to this is a vector space. And the reason for this, and this will become critically important in what we're about to do, is that if I think about this thing, and now I think about just this part of it. So I think about this piece and this piece. Well, there's an inclusion, right? This set sits inside here. And I'd like to think of that as a as a homomorphism of vector spaces, a linear transformation from R2 to R3. On the other hand, I can't really do much with 2 and 3. I mean, 3 is bigger than 2. But there's no other relationship. Whereas R2 can sit inside R3 in interesting different ways. And here, how should R2 sit inside R3? Well, this should go to this. Maybe this is one basis element, this is one copy of R, and this should go to this, and nothing goes to this. And that map tells you something about the relationship between these 2 spaces, okay? And this is, people, well, what this is called, and also how it's sometimes referred to in this literature, is functoriality, which just means you have maps between more structured algebraic objects. Okay, so great. So that's that's sort of a topologist analog of clustering. But now the point somehow is that you can do more sophisticated things. And so, for instance, if I have this rectangular, 2 rectangular regions, and I take one and I punch a hole in it, I can tell the difference between these things by looking at, and in general, I should say that one of the basic ideas in algebraic topology that all of this is riding on is the idea that you understand things by mapping in simple test spaces and looking at what happens to them. So a simple test space that can distinguish these you might think about mapping in is some idealized circle. And now if you think about what happens, well, if I map a circle into here, however, here's the image of my map of a circle, I can shrink this thing down to a point in a smooth way. If I map a circle in over here, well, if I map one in over here, I can still do the same kind of shrinkage. If I map it in like this, I can't. Right? There's no way to pull it closed on itself around that hole. Yeah?
Sorry, what do you mean precisely about the space that the circle represents?
Well, what do I mean by the space the circle represents?
Yes, I mean, it's not R2 exactly.
So this circle is maybe, a subset of R2 specified by the equation x squared plus y squared equals 1.
Just the path on the edge?
Yeah.
Okay.
Thank you, yes. So yeah, there'll be an important distinction between boundaries and interiors, and this is just the outside of the circle. Okay, so by mapping in the circle, I can detect things, and in fact, I can have, if you think about this, if you wind around a bunch of times in this direction, or you wind around a bunch of times in this direction, you might be interested in counting the number of times you do that. And so in fact, you could think about this invariance as being an integer, having to do with this map. But the point here is that I can tell these things apart. And now, let me say something a little bit about what this means to be qualitative and, yeah?
Just to clarify that, the first path you drew in the lower right, that would correspond to the integer 0.
Yes, that's right.
Because that one can be shrunk to a point.
That's right.
But if you wrap around n times, that would be the integer n, clockwise, and minus n if you do it counterclockwise.
That's right. That's exactly right. And so one thing to note about this is it didn't matter. If you think about it, it doesn't matter if this isn't a circle. This thing in the middle, any hole I make, I could make this hole. And I still have the same phenomenon, effectively. And so the point, so this is not sensitive to the specific details of the coordinates of the hole I cut. And where this came from, this is a piece of history, this area was invented by Poincaré at the turn of the century, essentially, in order to understand behavior of differential equations that were sensitive to global properties of the space or that the differential equation was living on. And so one of the salient points about what this is supposed to do is that it's supposed to be insensitive to changes like this. You really don't want to be able to distinguish between this and, well, I should make sure this is filled in, and this. Okay, so great. And you can keep doing this. Where am I? Let's say right here. You can keep doing this in the sense that this is a two-dimensional picture because it's what I can draw, but you could ask about spheres, right? And you could ask about, in general, you can ask about holes, n-dimensional holes inside of other spaces. And is there a hole, and how can I detect a hole? Where if I put a sphere in, does the sphere go around the hole? And if so, can I shrink it down or not? Okay, and that's what these invariants are measuring. Now, this discussion is all well and good, but in order to apply this, I have to get from a picture like this, from these points, back to something like that I can ask these questions about, right? And there's no interesting maps from the circle to 6 points. I mean, I guess there are 6 maps, but they aren't good maps. So the thing that I might do, and sort of the next move, is the following. So let's imagine here our idealization of the situation is that we have a metric measure space which is a metric space, x delta x, and it has a probability measure. Okay, so this is just an idea of I can sample from something that has a notion of distance. Okay, that's all this is. That's what this is a abstraction of. And so if I take a sample, what I get, I'll think of, maybe I'll write as m now, del m, I get some finite metric space. Okay, so this is a finite set of points with a distance. And what I'm assuming about the distance is, the most interesting thing is that the triangle inequality holds. Okay, and that's basically it. So, and in fact, and these are very weak assumptions in some sense, because I'm not assuming that this metric is continuous. In the sense, in examples of interest in biology, sometimes this metric is discrete. That there's a minimum, if you use the Hamming distance on vectors, for instance, there's a minimum distance between things. So one cautionary point, especially when reading this literature, but also when thinking about it, depending on your background and orientation, is that a lot of the insight and intuition people have for this comes from the idea that, oh, I'm sampling from a manifold. I'm sampling from a curved piece of paper in space. And I want to reconstruct something about that piece of paper. And that might be totally wrong. Right? And nothing about this assumes that what you're doing has any kind of smooth structure or any kind of nice local geometric structure like that. This is much weaker in some sense than that, those sorts of assumptions. But anyway, so we've sampled, and we have these samples. And I'll come back and say something about the sampling process, but for now we just have them. And what are we going to do with these points to produce an object that we can do algebraic topology with? Right, that's our job now. So I have these points. Right, so here's the idea. So so let's say that x, once again, is the circle. So we actually have a circle, and I use the volume measure on the circle, but any point along it is equally likely. And the metric is the distance. So distance between the points maybe is the distance along the circle, or maybe the distance, but maybe really I've projected it into R2. And the distance is the Euclidean distance. You have choices, and this depends on how the data is presented to you. So now I have this, and I would like to get back to the circle somehow. How do I do that? So the idea, and this is a very old technique in algebraic topology, which can be exploited for this. The idea is the following. Let's fix an epsilon. And now what we'll say is I will connect points xi and xj if the distance between them is less than or equal to epsilon. Okay? So let's say I picked epsilon to be slightly bigger than half the distance between these things. Then, I can draw these circles, right? Then this represents, all the points within epsilon of that thing. And I connect the edge. And if I picked epsilon just right.
Half epsilon or 2 epsilon?
Yeah, so there are different conventions about this. I probably want either of those would be fine. I mean,
the way you do it with the disks, it seems like you're connecting them if they're within 2 epsilon.
Yes. Yes, sorry. And thank you. Yeah. So if I pick this just right, then I get a graph. Okay, so I get a graph. And the graph, again, if I did this right, the graph has a cycle. If I didn't do it right, then there's some gap somewhere, right? Yeah?
If there are points that are closer, a bunch of points that are really close together but on the circle, they would still all be connected one to the other, or you're only connecting one to only one other?
No, that's right. So really, and this is the danger of drawing these pictures like this, really what happens is, right, what is your, even if I'm sampling from something like a circle, of course, right, I get something like this, and then I'd connect all these points. And so we'll come back to that, but yes. So, okay, so good. So now from this graph, well, it's a relatively straightforward algorithmic matter to detect if there are loops in this. Okay, so from this discrete object, I can now tell, does this thing have a loop? And maybe for each of these loops I can find, I choose a copy of R and I assign a vector space. Okay, and this is the basic. So this is sort of the beginnings of combinatorial topology or simplicial topology. Now, let's.
For the loops, you could assign for every connected component.
That's right. Yes.
That would be like your first example.
Yes. That should have been, thank you. That should certainly have been my first example. Okay, so, right. So we have the points. And I do this, and let's say I pick epsilon so that, that these ones are connected, but these ones aren't. And when I say pick epsilon, I mean something like you should imagine that the distance is here bigger than all the other distances, even though that's not how the picture came out. And now this thing has 2 components. And so you would assign to this your R2. And this has 1 loop. And so you would assign to this R. And this is often or I'm getting towards what people often call the 0th homology group. And this would be H1, which is the first homology group, or vector space. And I'm not going to, that's the language, or if you look at this literature, if you know this, of course you know that, but if you don't, that's all that means. Well, that's not all that means, but that's the beginnings of what that means. Okay, so so this is great. This was in dimension 1. So now you can continue this process, and you can play the following kind of game.
It's kind of working.
Yeah, this isn't bad at all. So now what do you got? Well, you can form what's called a simplicial complex. And the idea of a simplicial complex is that if you want to compute things about spaces, it's much easier if you have some kind of discrete model, some sort of combinatorial model of them. All right, so here, our combinatorial model of the one-dimensional part of the space is a graph. Graphs are comparatively algorithmically tractable objects for lots of sorts of questions about them. In a way that if I gave you, x squared plus y squared equals 1, that's much harder to deal with in some settings. Right. So a simplicial complex, the idea is the following. We're going to extend this notion forward by saying we're going to think about spaces as built from simple pieces. And the pieces look like triangles. So we have what are called 0 simplices, and those are points. So you get you can take as many points as you want as you're starting. And that's, well, I guess it's not there anymore, but that was here. And your 1 simplices, those are line segments. And one thing to note is that each line segment has a boundary, 2 points. And the way to think of this is that boundary tells you how this is glued to stuff. Okay, so how do I build a model of a circle? I take 3 zero simplices, which maybe I would give names, V0, V1, and V2. And then I take 3 line segments. And I take, here's, I don't know what to call this. Let's call that Z1, Z2, and Z3. And the idea is that Z1 is glued on the boundary to V0 and V1. Okay, so what this is it's an idea of building a space by giving very simple objects and gluing instructions, and a regular algorithm for how you glue. Okay, and in fact, you can imagine there could be some notational compression. I might just call Z V0 and V1. So I just have to tell you what it's glued to. It's a 1 simplex, so I have to tell you it's a segment, and I have to tell you what it's glued to. Now you'll notice that I'm not being careful about scale, about how big these segments are, and that's because we're working in a context, maybe as evidenced by that picture now erased, where we don't care. Right? We're looking at invariants that can't tell the difference between this being the interval 0, 1 and this being the interval 0, 2, let's say. We're just, Okay, so that's 1 simplices, and now we have 2 simplices, and that's going to be a triangle, so we'll have 3 points. And that has a boundary. What's the boundary? It's this, and this, and this. Okay, and so now I can, after I put in my 1 simplices, I can glue in some triangles. And I have 3 simplices, which are tetrahedra. So these are what is this? This is sort of the convex hull of these points, right? So this kind of data specifies a simplicial complex. And in the context of a talk like this, I don't really want to be a lot more precise than this. But the point of this is that from this combinatorial data, from this gluing information, so what do I have to do to specify a space? I just have to tell you how many simplices there are, and by their naming convention, how they're glued. And that's it. It's not very much data. But from this, I can figure out now questions about stuff like this. So for instance, in the way you do that, well, so how do I make a circle? So here's a circle. In fact, we have one here. I take this thing, which you should think of as 3 zero simplices and 3 1 simplices. So I took 3 segments and I glued them at their endpoint. And now I have, and that's a circle, right? So that's a model of a circle. You can go around, right? And we don't care about corners or things like that. We're not dealing with that kind of geometric information. There are other circles. This is not a unique representation of a circle. I could use as many segments as I wanted. And there are reasons to want to have finer versions. This is a very coarse circle in some sense, because if you think about this, all paths along this were probably multiples of 3, at least in terms of what you can understand from the simplicial structure, and you might want to have other paths. But anyway, we have this, and now how can I tell? So if I was assigning h1 to this, right, I would assign to this, r, because I have this loop. Now, supposing that if I took a circle and I filled it in, so I now have a solid disk, it's no longer the case that I can go around, that if I put a loop around the end that goes around that I can't pull it into the middle to a point. So this no longer has a hole in it, right? What's the difference between, and what does the combinatorial model of that thing look like? Well, I just drew it. It's something like this, right? Here's a model of a disk. What's the relationship between the disk and the circle? Well, this model of the circle is the boundary of this disk, sort of evidently. And the algorithm for computing homology essentially is asking for any possible hole, is there something that it's the boundary of, that fills it in? And that's easy to say, and at least in this picture, hopefully is clear enough. The sort of cool part about all of this is that you can solve that as a problem in linear algebra. So it turns out that you can set things up so that there's what's called the boundary map, which for each simplex in your thing, or for each sort of collection of simplices in your thing, tells you what the boundary is. And you can ask a linear algebra question about the behavior of that map, basically. So the point, and this is the sort of thing, again, if you know this, that's great. If you don't know this, what I want you to remember from it is the following stuff. So simplicial complexes are discrete models of spaces, and two, computing these topological invariants is linear algebra, right? So as one keyword, if you're interested in looking this kind of stuff up, basically what's happening is that you are putting the matrix representing a certain linear transformation in Smith normal form. Okay, so it's nothing very fancy. Now, you might wonder, and I'll say more in a little bit about the computational complexity of all of this. The computational complexity of this depends. It's essentially matrix multiplication dependent on the number of simplices. And so the issue always in this is how many simplices did you actually use in order to build your space. And that can actually be a problem, right? So supposing I do the following thing. Here's my set of points, and let's say I choose epsilon to be this big. What graph do I get? Right, the graph I get is the complete graph. Right, everybody's connected to everybody else. And that's bad, right? Because how many edges do you have? Well, you have an exponential number of edges in the number of points. Okay, so one of the big issues in applying this, and I'll say stuff about this, is that if you're not careful with your parameter settings, you can end up with even though the linear algebra isn't in itself particularly problematic, you can end up with an enormous number of simplices, even as a function of the number of data points. So one sort of piece of mandatory disclosure about the behavior of topological data analysis is that this is not useful for at least on its face, is not useful for big data. Because just meaning the largest data sets that I think you can reliably do this for, for reasonable epsilons, are probably now after many years of work, are maybe in the tens or 20 thousands of points. You can't do this on millions of points. I mean, people have sort of hand-picked examples that do work in larger data sets, but generically speaking, if you have more than a couple thousand points, you have real problems figuring out how to somehow subsample that or project it down in order to use this. Yeah?
So how does this mathematics handle things like a space-filling curve, right? Because I think that's kind of related to this idea of a bigger epsilon. You're getting a bunch of points, but if it kind of converges to like the higher dimension, does that exist in this algebra?
So some of that stuff you can't really see because you can sort of you're not. Two things to say. One thing to say is some issues like that you can't see because we're working up to these sort of deformations of stuff. Lots of point set topology badness goes away. So I think the real thing to say is that this is sort of like if you think about if you start in point set topology, which is just sort of questions about the behavior of metric spaces, basically, then you can see all of these really horrible examples. In this kind of algebraic topology, you tend to be in a setting where you can't distinguish, for instance, the topologist sine curve from anything else. And so you don't have such bad problems with that. You do, and we'll talk about this, we are going to have terrible conceptual problems associated to another class of perverted examples, so hang on. Yeah.
I don't know if you're going to do this later, but conceptually, though, this notion of just what is the dimension of our data in terms of if it were being drawn from some noisy manifold,
yes.
How does that factor into this picture you just...
Yeah, so there are a couple of things. So this is a good question about dimension. There are a couple of things to say. So one of them is that, and let me start by saying something somewhat related, which is that if you think about this process we did here where we connected edges with an epsilon, this should remind you very much of the first step in any kind of manifold learning algorithm. Right? What does manifold learning do, usually? And if you're a specialist, I apologize now for what I'm about to say. But essentially, all manifold learning algorithms work the same way, and all of them do something like the following, right? They take a point, and they fix an epsilon, and they take a bunch of points with an epsilon, and they take those vectors and use that as an approximator for the tangent plane. Right, and now they have little approximators of the tangent plane everywhere, and then they do some global optimization to align those. Okay, and that's manifold learning in the 30-second nutshell. Okay, so this is very much like what we're doing. I mean, usually they use some kind of kernel so that you use a weighted graph and you smooth out these distances a little bit so that the shorter they are, the more you trust them, basically. But it's a similar idea. And so in manifold learning, of course, the dimension is essentially the dimension of this little local tangent space. Now, in this context, the way this comes into it is essentially that you can tell, if you build a simplicial complex from this thing, then sort of, as you'll see in a second, because I haven't told you how to get a simplicial complex yet, but I will. And as you'll see from that, you can get simplices all the way up. So in some sense, you can get very high dimensional stuff. But really, the cutoff for how high we expect to see non-trivial homology information is often an estimator of dimension. That having been said, there are a couple of other things that can happen. One of them is that if you think about, even in these toy models of, I have a smooth thing, I have this piece of paper embedded in some Euclidean space, the ambient noise is often in the dimension of the space you're embedded in, not the intrinsic dimension of this thing. And so there are sort of the usual kinds of issues that arise about extracting your signal from higher dimensional noise. The other thing I would say is that this, what to say? There's no free lunch. The problems that come up in any kind of geometric, for instance, with dimensionality estimation that come up in any geometric context, any geometric algorithm, they come up here and they come up here in very similar ways. So, okay. So, right. So I should tell you now, I haven't yet, but I'm about to, tell you how to get a simplicial complex from this picture. I've told you how to get the one simplices, but I should tell you how to get the higher simplices, and of course, you do the same thing, right? So what's the same thing? So here we have, you connect these points. So how do I specify a higher simplex? Well, remember, for for these two simplices, I just have to tell you of these 3 points what they're connected to, and really, also what I do is, if these points are with an epsilon, then I put in edges, and now if I have a set xi, xj, and xk, so I have some 3 candidate points, I put in a 2 simplex if, if I have the edge between xi and xj, and I have the edge between xi and xk, and I have the edge between xj and xk. So if I have all the edges, then I fill it in. And I do this, and there's nothing special about 3, right? So the k simplices, these I guess the 3 simplices would be some sort of tetrahedron. These tetrahedrons now arise anytime you have 4 points that are within 2 epsilon of each other. And this has a name. This is called, what I'm describing is a construction called the Vietoris-Rips complex, which I might write as VR epsilon of x. Now, there are other things you can do. Right, there are many, you could sit here for the rest of the afternoon and cook up ways to assign simplicial complexes to data. But this is a nice one because it doesn't assume any geometry, right? In the sense that all you know about is the metric. So what's something you could do that does assume geometry? If you knew your points are embedded in Euclidean space, you could blow up balls around them, real ones. And you could ask if they all intersect, right? This is just pairwise. In some sense, this is a pairwise condition, but you could ask for the whole set of points that, rather than asking, so if I have these 3 points, I'm asking, do I have this simplex? Do I have this simplex? Do I have this simplex? But instead, you could do something like this. You could draw this circle and this circle and this circle, and you could ask, is there a point in the intersection of all of them? And these 2 things might be different. This is what's called the check complex. And I guess this is the Vietoris-Rips. But in order to ask questions like this, you have to be able to compute the intersection of a collection of k-dimensional balls, which in of itself is sort of an interesting algorithmic problem, and also only really makes sense if you imagine your data is embedded in Euclidean space. And maybe you want to do that, and maybe you don't.
One quick clarification that might help is maybe can you draw the square as an example? we're not going to necessarily fill that in, right? You said a triangle on the B surface would always get filled in, but the square would not necessarily get filled in.
That's right. Yeah. So with the square, we could have something. You know.
Right.
Those aren't so. You know, you imagine those are idealized circles, but the point is that you're missing these points in the middle.
So we can still have a hole, is my point.
And so it's in the check.
In both. The triangle you can't.
Oh, the triangle. That's right. That's right.
Oh,
That's
with
right.
the-
It
with
was
the
a square.
square,
That's right.
you still
Yes.
can.
Yes. 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 Thank you. That's
Or
right.
the circle you drew before with many, many points.
That's right. That's right. Okay. So great. So what we do, you know, what is the first cut at our invariance? So where are we? Maybe we're here-ish. Okay. Yeah, I think we are. Great. Okay, so our first cut at our invariance is just you take VR epsilon of x and you ask, you know, what is h1 of that? Right, where h1 again is a vector space whose dimension counts the number of holes. Okay? That's a topological invariant of data. Now, here's a problem that you know, should be evident, and I alluded to, which is that you have to choose epsilon properly, right? So let's think about this picture again. If epsilon is very small, what I get is dust, right? I get just these points, and there's no geometry. Epsilon is close to 0 relative to the feature scale of this thing. If epsilon is sort of chosen correctly, then I should get a circle, something like a circle. It'll probably have extra, you know, as discussed, it'll have extra little simplices, but that's fine. If epsilon, you know, so epsilon equals 0, we have dust, and we have h1 equals 0. Epsilon is, you know, right, h1 equals r, and we have, you know. Now, if I choose epsilon to be, you know, the size of the blackboard, then once again, I get the complete graph. Everybody's connected to everybody. And in this case, h1 is 0 again, and you might call this heat death. Okay, so I have to know what epsilon is, and the problem is worse than this, right? This is you know, this is everyone's favorite toy example. Maybe not favorite, but it's the easy toy example to draw. But it's a misleading bad example, because of course, if you're really working with even something that's really geometric and nice, this is a silly thing to imagine would work, because look, supposing I have this thing like that, and I sample it at different scales, let's say. I sample the two bulbs at different rates. Or even if I don't, one of them is bigger. There are different epsilons at which you expect to see this circle and this circle. Right? So you can't pick a right scale because that question doesn't make sense. And things could be worse, right? You know, in some sense, there might not be a scale at which features exist. That idea might not be right. So what do you do about this? So there's no right epsilon. So what do you do about this? So the big idea, and this comes back, the reason I wanted to emphasize this point about working, I guess this was here but now erased, this big reason I wanted to emphasize this point about working with vector spaces is that the big idea is now you take all the epsilon. Right, this is the point of departure. When people talk about topological data analysis, this is the main point of departure, is that you take them all. And what do you do? The point is that if you have vector spaces, well, if I have epsilon 1 of x, so I have the thing for epsilon 1, that sits inside the thing for a bigger epsilon. Right? If you have a bigger epsilon, I've added more edges. So it's strictly a subset. Okay, so this includes into VR epsilon 2. What you might say is that what I've described is a functor on the poset category associated to the real numbers, but I mean, all I'm saying is this, right, for epsilon 1 less than epsilon 2. And now, well, what do? I track what happens, right? I track the bases of these things, right? So remember, I had that example a while ago with the connected components, you know, but let's do it again with circles. Let's say that in this let's say I have, you know, these are very densely sampled. Those are not so densely sampled. If I think about what's going to happen to the homology, Well, at the beginning, there'll be nothing. Let's think about time here. In the beginning, there's nothing. Then I see a circle appear for this thing. And so I'll draw that. There's a basis element associated to the R corresponding to this guy. Then this one, at some point, comes into things. Probably, perhaps before this one closes. This one's a little bigger, so slightly before this one closes up, this one closes up. And so that basis element disappears again. This basis element goes on for a little while, and now it's done. Okay, and here's what this is, and this is a summary of the change as epsilon changes in these vector spaces. Okay, and this is what's called a barcode. You're parameterizing this in terms of the endpoints, and so you could also draw this if you wanted. Well, there shouldn't be 3 of them, but anyway, as points in R2. And these are the basic invariants that people work with. So, and this idea, I guess I should give this a name. This is called persistence. And we talk about persistent homology, which just means tracking what happens to these vector spaces as time goes on. And so now this has so now you can see things at different scales. And you might also imagine, this is not always true, but a motivating idea is that if you have some little teeny tiny things, those are possibly noise. Now, one of the examples of the application of this that I like the most, and not just because I was involved in work on it, is that actually is one where even the little tiny bars are an important signal. So it's not always true, but it's a reasonable intuition in examples like this one. Right, because where would these little teeny tiny bars come from? Well, maybe there's sort of an artifactual circle that turns up right there. And maybe you didn't want that. Maybe you did, but maybe you didn't. I mean, in order to, again, as with any geometric algorithm, you really have to have some idea of what the feature scale of your data is in order to understand how to interpret what you're seeing. Okay, so the big theorem, let me state the big theorem about this, and then I'll get to the bit about what to actually do with this. Or how to start thinking about what to do with this. The big theorem about all of this, there's sort of two big important theorems. One of them is that this kind of thing is a complete invariant that really does describe completely how these things change, these pictures, and moreover that you can compute this fairly effectively. I mean, subject to these issues about the number of simplices, but given that, you can compute this. And this is, I haven't really been writing names of anybody, but this is sort of due to Edelsbrunner and Zamarodian and Carlson, and probably I should mention John Harrer. Now, the other big theorem in the subject is that this invariant is stable in the sense that if you perturb your metric space a little bit, so I'm going to state this non-mathematically because I don't want to introduce the machinery to give a precise statement, but let's just say that small perturbations in your metric space, your input, give rise to small perturbations in the output, in the barcode. And the point is that you can think about a metric on barcodes. What do? You permute them around and you match them, and you look at the excess. You have some earth mover type distance, for instance. You can think of it that way. And of course, it turns out there's also a metric you can put on the set of metric spaces. This is what's called the Gromov-Hausdorff metric, which is great, but I don't want to describe now. And sort of what this says is that if you have a small change in the metric space in the Gromov-Hausdorff metric, you have a small change in the barcode in a suitable metric on barcodes. Okay, so that's nice. Great, so that concludes the very brief overview of what this stuff is doing. This is not, by the way, I should say, there's another kind of technology that came out of these ideas called Mapper, which you might have seen. I'm going to say nothing about this. I will just simply comment that is the technology that was commercialized at the company, IASD, that Jonathan alluded to, not so much this. But I like this better, so I wanted to tell you about it. So now, here's the question. There's sort of, you know, how can we use this? And there are three sorts of questions. I think there were three that I wanted to articulate. Yeah. Some of them are questions about estimation. Some of them are questions about robustness. And the most interesting questions are about inference. So let me tell you about these in order. So estimation, the question here, these are questions of sort of very basic statistics. You know, supposing that I have something I can sample from, and I sample a lot of points from it, and I do this, does that approximate the support of this distribution? Right? Is there something like a law of large numbers in this context? And the answer is yes. Okay, so the asymptotics, so one question is, you know, can I approximate? Let's assume the support of this is the whole thing. So can I approximate the persistent homology of X from persistent homology of Xi, where Xi is a large sample? So that's a very basic kind of consistency. And there's been, you know, and the answer is yes, you can. And there are various kinds of estimation questions you can ask. One thing to say about this is that the kind of statistics you're doing here is hard. And it's hard, well, or the things people do are easy. But the reason the things people do are easy is mostly because doing anything sophisticated is hard. And the reason is that where are you doing your statistics? Right? You're doing your statistics on the collection of these things with some weird metric, right? That's your space. And it does turn out, you know, it's a complete separable metric space, so you can do probability theory, but it's a very poorly behaved one. And you might ask, what is the average of a barcode? Right? This is a very reasonable question to ask, and there's not a good answer to that. There are notions of centroids in any metric space, but this metric space is one where there aren't unique centroids. So what you mean by moments of a distribution is kind of fraught. So, and if you try to think about the behavior of stuff like the central limit theorem for these sorts of things, it's also complicated. So it's hard to do, like the things you want to do are hard, and so the statements that you can give are relatively basic. Now, one way you can deal with this is, you know, and one of the things people do is you pick some function. Let's call this, let's give this a name. Let's say that B is the set of barcodes, or the space of barcodes, I guess the metric space. And you can think about various families of functions, theta, from B into R, or maybe more generally into Rn. And so you know, what do? You say, what's a function? I'm going to count the number of barcodes. That's a function. I'm going to take the maximum of the rightmost endpoint. That's a function. You know, again, we could sit here all day and cook these up. And now when you have a function like this, well, you can do statistics here. That's great. We know how to do that. You now have these troubles, right? That if you average over here, it doesn't mean anything, right? This is not a surjection. You don't get everybody, and you can't go back typically. So what you do with this is sort of hard. A related issue is one of robustness. And, you know, here, and this gets back to this, I was promising perverted examples earlier, and now here they are. So if I'm not careful, you know, if my data, what I'm working with, looks like this, but, you know, out at infinity, I add some little tiny circle, I'm going to pick that up. Right? So this is not a robust process in a precise sense. And what I mean is that by adding a small constant number of points far away from everybody else, I can change these invariants arbitrarily. I mean, you know, it's like the mean, right? I mean, we know lots of examples of estimators like that. It's not, and there are ideas about how to deal with them. But it's a thing to know that it's particularly pernicious here. You know, you can think of you know, adding endlessly complicated, tiny little geometric objects and make these things whatever you want. And then that heralds, but there's still, you know, so what do? Well, you could resample, and you could look at, you know, the resampling distributions, and that's a way to get around that kind of stuff. I mean, the usual things you do are the things you might try here, and they work with varying degrees of success. But maybe the point that I really want to emphasize is that inference is kind of a disaster. And what do I mean by a disaster? Well, usually when you want to do, when things are easy, you have an idea about what your generative hypothesis is, right? You have a parametric family of distributions, and you're trying to estimate some parameters about them, right? You might hope that you live in such a world. And here, good God, we can't specify anything. It's algorithmically intractable to even imagine, there's sort of famous algorithmic intractability results about trying to imagine classifying or describing classes of spaces up to homeomorphism, up to any kind of sameness you want. So you have no hope of, here's the question, right? You give me this data. You worked in the lab and you got this data, and I did this thing, and I say, oh, look, it's got R7 in the 6th homology group. Okay, now what? And that doesn't, that constrains nothing almost about the geometric object. What do you do with that stuff? So one thing you can do, of course, what does everyone do? This can be a stable, uninterpretable feature for deep learning, right? You can feed this into a neural network and hope something comes out. You can do this. I don't find, as an engineering matter, I imagine there are cases where that's useful. I don't find that scientifically satisfying, personally. So when can you do inference in a meaningful way? When can you use this stuff? Given that statistics is hard and specifying hypotheses is hard, well, the answer, I think, and I should say that I'm not delivering received wisdom. I'm something of an internal skeptic, and I'm telling you sort of, my jaundiced and dyspeptic view of this. Nonetheless, I like this stuff a lot, but I think that the reality, it's important to be clear-eyed about the things you love. So when is this going to be good? It's going to be good when you're working this is when geometric data analysis is always good, right? When you're working in low dimensions and you have very clear and constrained geometric hypotheses. Now, to be fair, let me say two things. I'm sort of rapidly running out of time. One thing to say is that it's a little bit unfair to knock topological data analysis for having these constraints. In some sense, you always have these when you do any kind of geometric data analysis. People can get away with, PCA you don't really have these because what you're doing is so terrible and crude, or anything like that. Most of the dimensionality reduction stuff people do is like slam something into R2 and hope there's some structure that survives, right? And you probably should have done this anyway, but you can kind of get away with not, because what else are you going to do, right? But here, because it's a more sophisticated tool, a finer instrument, you are forced to confront these issues. And I don't think that's a bad thing. It makes you think clearly about what you're doing. And and there are cases where this actually is true and this is useful. And now in 3 minutes, I will tell you to go read the paper, basically. But there's one example of this that I like a lot is that you can use this. Imagine that what you're doing is you have genomic data that you're imagining is going to be fit to trees, right? So you're going to make little phylogenetic trees from a finite metric space, and you want to estimate recombination rates. Now, you might imagine that if your points are consistent. Oh, that's not really a great picture at all, because that's not what's happening. Because really what you're seeing are the leaves. But yeah, anyway, if the points you have are consistent with a tree, well, that's not going to have any H1, right? There's no circles in that. It just all shrinks back to the root. If there was a recombination event, maybe you might hope you would detect that by a circle. And so it's not crazy to imagine that looking at counts of H1 tell you something about this. And in fact, it turns out that looking at rank counts of H0, persistent H0 for these, the genomic distances between these leaf points, actually lets you estimate the recombination rate. And this is a nice example where, and if you work it out, you can give coalescent theory interpretations of what these quantities mean in idealized cases. So we actually have an idea about what this is doing. We have a real, you have a real hypothesis, which, as with all real hypotheses, is more complicated than in practice than one hopes, but you do. And you can actually get something good out of this that I think is hard, has advantages over other ways you might get it. So I think the net, maybe the thing I want to leave you with is that, they're really, what do I believe anyway? Leave you with some religious views. There really is shape in data, and these invariants really do see it, but to do stuff with it, in part because of the statistical difficulties and in part because of the fact that there are a lot of spaces out there, you need to be in a setting where you understand what you expect to see, which is like anything else, but it's particularly sharp here. So anyways, that's probably a great place to stop. Thanks again for the invitation, and thanks for turning up. Yeah, so I should say this is and now for something completely different, and it's actually true in the sense that this talk doesn't. If you saw the first talk, great. If you didn't see the first talk, this doesn't have anything to do with it, basically. So that's maybe good for you. So what I want to say now is about, and this is what I'm describing, is joint work with Luis Aparicio, Mykola Bordyuh, and Raul Rabadan. And the idea is to use, let's say, use the results of random matrix theory to do high-dimensional PCA on single-cell data. Okay, and I should say as another mandatory disclaimer that whereas I don't know if I'd say these things on camera, but whereas I am in fact an expert on algebraic topology, I am not an expert on random matrix theory. I suspect there are people in the audience who know more than I do, but I will try to tell you the things that I understand about it as they are germane to this talk, and to those who do know, I apologize now if I say anything.
And actually, one side point, on March 13th, Alex Blumendahl, who is an expert on random matrix theory, is going to give a primer just on that subject, and then Christina Chen is going to talk about applications to genetics. So you'll have an opportunity for a full primer on this in a month.
So the point of departure for this is the following thing. So we have this matrix of, maybe I shouldn't say we have this. We had many matrices of single cell data, which is, some. Did you write a bit bigger in? Yeah, well, maybe. Let's see, how do I want to do this? Yeah, so we had, some n by p matrix where we have, and let me just see if I get this right, we have n cells and p genes, and the entries are expression levels. And if you do the following numerical experiment, right? So you start as if you're going to do PCA. So let's call this thing x, and you do x transpose, and you, I guess, divide in this case as I've set it up by p. So we take the covariance matrix. I'm going to assume, tacitly assume that this stuff has been centered so that it's mean 0 and variance 1 and stuff like that. If I forget to say that, I probably meant it. And if you do this, and now you do stuff like you plot a histogram of the magnitudes of the eigenvalues, okay, which is a reasonable thing to do. So you look at the spectrum of this matrix, right? So you get the singular values, which is what you would do. Then what you see is some sort of and what we saw across a wide array of examples was the same sort of pattern that looks kind of. Make sure I draw this right. It looked kind of like this, and then it had a few little bits out here. Okay, and this is magnitude, and this is frequency. Okay? And these are the eigenvalues. So this is, yeah, I guess I say that. Okay, so what is this? What are you looking at? Well, the observation is that, so you can see this. I should say one other thing, which is that if you plot, and you sort of get this characteristic picture, and if you plot, you know, something like the distances between the eigenvalues, then you get another sort of characteristic picture that looks maybe something like that. And the thing you notice is that, one of the things you notice is that this always seems to happen across a wide array of data sets. And the other thing is that these distributions that you're seeing, these shapes, are very well known. Right? This, what you're seeing here, is what's called the Marchenko-Pastor distribution. And this maybe is the, you know, Wigner-Surmise distribution. And these distributions, what these are telling you is what you are seeing, what this tells you, these distributions are sort of at the core of random matrix theory. Right? The main result, and I'll summarize the main results of random matrix theory in a second. But the thing to say is that what you're seeing is that the single cell data looks like most of it was drawn randomly, and you know, and you're seeing sort of the spectral signature of that. So the idea of the algorithm that I want to describe is one that uses explicitly what we know about or what people have discovered about random matrix theory to, in some sense, filter out this stuff and focus on this stuff. Because, as you might imagine, this stuff is noise, and this, ideally, and this artifact, this stuff out here is coming from a signal. Okay? And that's the idea. So it's some kind of dimensionality reduction. And there are, I should say that sort of the first cut at this, as I'll say, is pretty vanilla in the sense that this idea, you know, of using random matrix theory or ideas like this to do dimensionality reduction, or at least to do estimators for the behavior of the largest eigenvalue or something like that, this has been around. One thing that's kind of cool about, in addition to the fact that this actually works in single cell data, one of the things that's kind of cool about what came up, yeah, maybe this says something about me because this is the part that I think is cool. But the other thing that's kind of cool that came up in this example is interesting theoretical questions which are not well understood, having to do with sparsity in the data. And I'll say something about that, and I think that's sort of particularly interesting. Okay, so let me say something about what random matrix theory is doing now that I've, I want to say anything else about this? Yeah, now that I've drawn these pictures. And so the TAC, I mean, there are various entry points for random matrix theory. You know, one of them is, I mean, the most basic question, of course, you can ask is if maybe I should write this, you know, sort of a basic question. So I, you know, draw a random matrix. What are its eigenvalues? And maybe another thing to say is, what are its eigenvectors? Can you say anything about them? And I want to look at this through a slightly different lens that's germane to what we're doing, which is, comes from this kind of picture, which is the following. So suppose I want to do high-dimensional PCA. And I should say that this perspective, sort of the big exponent of this, is Johnstone. And Johnstone has a very nice, if you're interested in this stuff and you don't know this, the kinds of things I'm saying, Johnstone has this beautiful expository lecture he wrote for the ICM, which does a nice job of laying out some one way of thinking about this. But suppose I want to do high-dimensional PCA. And I'm in a regime where, you know, there are different things you can imagine. One thing you can imagine, and the thing that's easiest, is to imagine a regime in which the number of points runs off to infinity. right? So we have this n by p matrix, and I can imagine that n is going to infinity, as this thing is getting big, n is going to infinity much faster than p. And in that case, everything is really nice, right? In that setting, so if n goes to infinity much faster than p, then what you discover, the kind of thing you prove is that these sample, you have obvious sample eigenvalues and sample eigenvectors. And what am I doing, right? I'm drawing random covariance matrices, right? So that's what's going on here. So we're drawing random covariance matrices. And if I do this, then these things are good estimators of the population eigenvalues and population eigenvectors, like I should say, are good estimators. Now, the situation, however, which is relevant in lots of examples, but is particularly relevant in the single cell data, is one where this is not the right asymptotic regime. Right? These are not the asymptotics you have. The asymptotics you have are something like p over n is going to a constant lambda, gamma. So that's what you see. And now the question is, what happens here? And to study what happens here, the first thing you might ask about is what happens if I take a random covariance matrix and I assume this sort of thing? What is the asymptotic behavior? And the theorems, and what you have is interesting and goes under maybe the heading of universality phenomena. And it's very cool, right? What happens is that it doesn't matter what distribution you sampled the entries of your matrix from very much, except for knowing the first couple of moments. So if you know the first 4 moments or something like that, then what always happens is that you have a characteristic. If I do something like make this kind of histogram, you have this characteristic distribution and let me maybe write a formula for that. This talk isn't going to have that many formulas, but perhaps I can write one for you. This looks something like, the density for this is square root of, let's say, C, how do I want to do this? C max minus x times C min minus x, and divided by 2 pi x times p over n. And now here, well, let me do that slightly differently, just for consistency.
Is that the first point for the single sample, or both?
I'm writing the density of the Marchenko-Pasteur. This is mostly irrelevant, except, it's not irrelevant, but I don't expect this is not going to play a role in the talk, and I don't expect you to remember this if you've never seen it before, but I want to write the density because of the behavior of these two constants that turn up, which I'll define in a second. So this is going to be gamma, and now the point in here, C max, is equal to, and let me just get this right, this is 1 minus, it's 1 plus root gamma squared, and C min is 1 minus root gamma squared. So this is just this parameter. The point here is that this parameter gamma is controlling this distribution, and what it's controlling is the width of the distribution, basically. And this gets called this is what's called the bulk in model, for reasons we'll see in a second. But moreover, so you have a distribution for the histogram of the eigenvalues. Moreover, you know distribution of the eigenvalues, and you know things about the fluctuation of the largest or the k largest eigenvalues themselves. So you understand the fluctuation of, let's say, the largest eigenvalue. And you know something more interestingly from our purposes. You know the eigenvectors are what often gets called delocalized. And let me say something about what that means. So the point is that, what do what you expect the eigenvectors to do maybe, or what might you hope, is the eigenvectors are basically, randomly distributed around the unit sphere. You normalize them, right? They're just all over the place. And if you look at their entries have a Gaussian distribution. The energy is smeared out all over the eigenvector. Okay? So you get sort of, the eigenvectors are non-informative in some sense. And they should be non-informative, right? I mean, you would hope this because you're drawing random data. Okay, so the idea is now to use these characterizations to, remove the noise from the results of PCA, right? And, okay, so in order to do that, you want to know what happens if I mix my noise. So great, so I've now sort of told you that there's this theory which gives you, with very weak assumptions on the distribution, tells you something very precise about what the spectrum looks like and what the eigenvectors look like, okay? And now I want to, use these characterizations to remove the noise from the results of PCA. So I have to do, so in order to model this, what do? Let me pause for a second and say one of the reasons that we were enthusiastic about this for use in single cell data, or in general, is that this doesn't make any assumptions other than these moments about what the noise is. Right, and that's good because, well, you don't know what the noise is, right? And if you think about, there are various ways to deal with, and I'll say something about this, with the issues of noise that come up in single cell data, and many of them make distributional assumptions. I mean, they're justifiable distributional assumptions. I'm not trying to criticize anyone. I would make them too. But they make distributional assumptions about where the noise comes from. And it's nice if you don't have to do that. I mean, not making choices has costs, of course, but but that's sort of one of the ideas about why you might want to try something like this. Okay, so what now are we going to do? So now we study, and this again, this terminology is due to Johnstone. To model this, you study something called the spike model, and the idea here is that now I adjust my covariance matrix. So I'm going to add a low-rank signal. I'm imagining I'm going to have noise with a low-rank signal in it. Okay? So I have sort of k large eigenvalues, lambda 1 through lambda k, and then I have the rest, and maybe I fix the rest to all be 1 or something like that. And this is the planted signal of rank k, and this is some huge matrix. Okay, so that's the spike model. And then the question is, what happens to this kind of distribution, and what happens to the to the eigenvectors if you do this? And the interesting thing is that you might, so let me write the question. For the eigenvalues, the thing you might expect happens. Right? What does that mean? The eigenvalues, if there's a threshold, and let me write the names for this. Let me try to do this correctly. Yes. Oops. Coming from Bake, Benaru, and Pesh, or BVP, there's a threshold, and basically, if your eigenvalues, so what could happen, right? If these eigenvalues are very close to the rest of the eigenvalues, then you can't see them. And what you get is, this. If the eigenvalues are much bigger than the threshold, then they kind of pop up. And the further away from the bulk they are, the more information that the eigenvalues you measure recover about what you planted. So if lambda k is much larger than, let's call this the bulk eigenvalues, much larger than the bulk eigenvalues, then you can recover them to some degree from what you see, and they sit way out here. If they're close to the threshold, there's very interesting theory about what happens. So most of the, from a mathematician's standpoint, what's interesting is when these eigenvalues are not that much bigger, right, or close to this there's a phase change, basically, and it's close to this sort of phase change point. From our standpoint, that's not, we're less concerned about that. What we're concerned about is that if they're far away, then you can pick out the signal, right? And the extremely intricate and wonderful mathematics about what happens right near the threshold, for this particular application, or for this kind of thing, maybe we're less interested. Maybe you are, right? Because you actually would want to know for those things near the threshold, are they signal, are they not? But to first approximation, maybe we don't worry about that. I'm sorry?
I spent years of my life.
Yeah, I know. It's great work. I don't mean to. Okay. So now there's a subtle. Okay, so the first thing you might say is, great, we're done. We're going to pick off these eigenvalues, take those eigenvectors, run with that, right? And there are two things that are wrong with doing that. One thing, I'm not going to say any, well, I'll explain and then not describe how to fix, because I'm not sure, in the kinds of assumptions we want to make, I'm not sure you can, and it also seems not to matter too much. And one of them I will talk about how to fix. But let me talk about the two problems with the naive approach. Okay, so naive algorithm. Take the eigenvectors that correspond to the eigenvalues that separate from the rest. Now, there are two problems. One, those eigenvectors are bad estimators of the signal. And two, the matrix, the input matrix, is probably sparse. So let me take these in turn. So on the one hand, if you do this and you look at those eigenvectors, they look localized. And what do I mean by localized? They don't look like, the energy is not smeared out across them. They don't look like they were just kind of drawn randomly from the circle, from the sphere. So it looks like there's signal there. And there is signal there, but it's not the case that these things converge to the directions that you planted.
Sorry, when you say localized, you mean the number, the sort of non-zero coordinates are small? There aren't that many non-zero coordinates?
Basically, yeah.
Basically, like up to noise?
Yeah, so what you do is, what would you measure? You do something, people use this measure called the participation ratio. So you do something like sum over the fourth power of the entries, or something like this, in order to see if, again, what you're looking at is that whether or not sort of you expect whether or not the average, or the generic entry has magnitude that's more or less proportional to its share of the norm of the vector or not.
You do need to be at a basis where the signal is sparse,
in order to
do it that way, right? To be.
That's true. Yeah, that's true. So one thing that you prove is that these eigenvectors end up rotated. They have a non-trivial angle with the actual eigenvectors, and even no matter how many points you have, and that's bad. So I should say that this is the point of departure. So this is proved by, the nicest statement any of this was proved by Paul, and maybe there's work of Johnstone and Paul on this. I shouldn't say nicest. The first statement I learned. Because I don't know this literature as well as some. And this leads you, this is the thing you do based on this is sparse PCA. Okay, this is where sparse PCA comes from. Because if you make sparsity assumptions about, not sparsity assumptions about the matrix, but sparsity assumptions about the basis in which you can represent these things, then you can prove you can get a consistent estimator this way for these guys, okay? We don't want to do that. So I did do that when I was was just testing how this worked, and I can tell you how it went. But we don't want to do this here. And so we're essentially going to ignore this problem. This problem, two things to say. The eigenvectors aren't great estimators. The eigenspace is a slightly better estimator. And you can get, and if these things are large enough, it gets close enough that for the kinds of things that we're doing, it turns out to appear not to matter that much. But this is a real problem, and one that in the way I'm presenting this, we don't have a great story about. Now,
when you say the eigenvector is not a good estimator, but the eigenspace is, do you mean taking
it's still not a great
taking
estimator.
this set of
Yeah,
eigenvectors and looking at the space they span
That's right.
and that space?
Yeah, there are different notions of consistency. Like there's for each eigenvector whether you can recover the original eigenvector, and there's stuff about whether you can recover. There's span consistency, which is another useful notion. Those have slightly different properties. I believe so. But the real thing, again, I think the honest thing to say is that this is just a problem which we chose to ignore, and that turns to work out okay in this. But you should be mindful of it. The other thing to say is that the input matrix is probably sparse, and this is a problem that we were much more concerned about, or we were particularly concerned about, because single-cell data does have this issue, right? The matrices are sparse sometimes and so a next kind of question to ask. So, sort of a next question. What about sparsity? And here, what you see, and this, I think, at least my understanding, and you should correct me if this is wrong, is that here there's somewhat less theory than there is for sort of generic random matrix theory. But what you find is that there's a threshold effect, once again. So if you start randomly deleting entries from columns, then if you don't delete very many, nothing happens, effectively. But if you delete enough of them, then what you see is, sort of if the rate of deletion /sparsity is high enough, then what you see is that the eigenvalue estimates are distorted, or the behavior of the eigenvalues is distorted from what you might expect from this Marchenko-Pasteur distribution, and you see spurious localization of eigenvectors. And what I mean by that is I take a random matrix. Now I just throw away some of the entries, or maybe enough of the entries. And what I see suddenly is that it looks like there's a signal because many of the eigenvectors appear to point in a particular direction rather than being smeared out. Right? So you expect that the eigenvector that it should look normal, right? And instead it doesn't. And we want to control for this. Right? And this is the big problem when you think about the algorithms people have for dealing with single-cell data. There are various ways to deal with this having to do with imputation of entries. Right? That's one big school of thought about how to do this. And we did something slightly different, which is just to throw out the genes that are, our approach to this is to throw out the genes that are sparse. So let's do this over here. Let me see if there's anything else I wanted to say before I say that. Number 2. Yeah. So the idea is this. to control for the effect of sparsity, we randomize within the column. So for each, we randomize within each column, you randomize the cells. So you're trying to remove all trace of the signal, but you are retaining information about a particular gene. And then then you find the eigenvectors of the covariance matrix, and you project onto them. So you look at the representation on the basis of eigenvectors, and for a gene that doesn't look like this, if it looks like it's got some preference in that eigenbasis, we throw it out. And there's various, you could say a bunch of things about how to choose the cutoffs to do that, but that's, at a high level, that's what happens. So you delete these genes that appear to be inducing sparsity effects. And now, and so now what happens, so this compresses your matrix a little bit. And so what is the procedure? So the procedure is now you do this reduction step to remove sparse genes. Then you plot the spectrum and fit a Marchenko-Pasteur distribution to pick off this part. You retain, yeah.
But one gene at a time? You shuffle this out?
You shuffle all of them, but within each column in an uncorrelated fashion. So I think the answer is yes. Log counts. So you plot the spectrum. You pull off the largest eigenvalues and then associated eigenvectors. And now, what do you do? Well, now you can play all sorts of games. So one of the kinds of things we did is that you project the genes onto the signal eigenvectors, and you project them onto the other eigenvectors, and you preferentially select genes which are mostly concentrated on the signal part of the basis, right? And there are various ways you could do that. And then you use that, and the validation tests we did are, then you cluster, right? And the kind of thing you're doing is you're trying to pick. We have these data sets where, for blood cells or things like this, where people know various cell subtypes, and you'd like the clusters to reflect the cell subtypes. That's so you have this sort of supervised learning task in some sense. I guess, well, we're not treating it as supervised learning, but you have this ground truth that you're matching against. Yeah.
Sorry, so what's the actual procedure? You take just the top of the singular value decomposition, you project to the largest eigenspace?
So one thing you could do is project to the largest eigenspace, yeah. Another thing that we did was first subsample the genes for the most informative ones and then project to the largest eigenspace. Further pruning of the genes based on the ones that are mostly expressed in the largest eigenspace. Yeah.
So, okay, heuristically, you're telling me that this works to achieve the task of making the clusters that agree with the truth, better.
Right.
If you do it naively.
Right.
But partly, I want to know, is there a theorem here? So imagine we have an algorithm which isn't just this hard cutoff at a threshold, but we're doing something like randomized SVD where we preferentially keep genes that are less sparse. And we just run this to convergence, and then we prove a theorem that says, how what it converges to should relate to
I think
the truth better than if we did
right.
it with equal weight on all of
Sure, and the honest answer is I don't know. It looks plausible that there could be such a theorem, but I don't know. There are a bunch of things to say. So, the results of doing this turn out to, this turns out to work well. And it works well compared to other things people do. Now, you could ask questions like, how does this work? I mean, there are a lot of ways. One thing to say about this is there are many, many ways to extract low-rank signal from matrices, right? That's a, and you might ask, how does this compare to sparse PCA? How does this compare to any of the stuff coming out of the compressed sensing literature, right? There are lots of low-rank estimators out there, and the thing I would say about that is that I think that you get, it seems like anyway, that you get qualitatively similar results from sparse PCA. Now on the other hand, one nice thing is that this is much faster than doing sparse. You're not solving some non-convex optimization problem. You're just computing eigenvectors, basically. You're computing a singular value decomposition. So there are some advantages over sparse PCA in that sense, and I can't claim to have done an exhaustive search. Sparse PCA makes further assumptions about the existence of a sparse basis, and I think and I don't know the answer to whether those assumptions are justified, but I think that's a thing one would want to know. In terms of doing the sort of low-rank estimation stuff, sort of a lot of that seems to have trouble with sparsity, but there's also a lot of work on how to deal, more with sort of sparsity on the other side, but some with sparsity in the matrix. And I think it's, I'm not sure I want to take a position on camera about whether or not you could do similar things with that.
So I think 2 points of interest for this community are going to be around, right? You've sort of contrasted doing PCA against versus sort of sparse or efficient sparse PCA in this fashion. And I don't think anyone's going to push back that, okay, this seems better.
Right.
But they may say that, well, okay, but PCA kind of sucks.
Right.
When it comes to interpretable dimensional reduction,
right.
PCA is garbage.
Right.
And we want to do things like NMF or graphical models, which have structured, Bayesian formulations. And
yes.
we want to do inference and so
yes.
on. So do you think that these ideas extend in those directions?
Yeah, so I think, there are lots of direct questions you could ask. So one question you could ask is, you're doing NMF. Do you see phenomena like this? Can you use ideas like this to analyze what comes out? And that's something that, that we're actively thinking about. It looks like the answer is, for NMF anyway, is yes, although it's less clean. But it looks like the answer maybe is yes. And you can also ask questions, and this maybe does relate back to the talk I gave earlier. So one thing you could ask about is, what about random graph Laplacians? Supposing you were going to do manifold learning, could are there a universality phenomena that's germane to that? And there, again, the answer is yes. It is the case that people, and then there have been at least a couple of nice papers on understanding what the spectra of random graph Laplacians look like, which you might hope could help you use ideas like this for more sophisticated geometric algorithms. So I think the answer is that it's quite plausible that you might be able to use, that you should be able to use this in more sophisticated geometric settings. But I can't say something specific.
I think there was a point that I didn't appreciate. So does this procedure remove all types of sparsity? Like, I can imagine that I have a gene, I have like 20 genes that across the whole data set they're sparse, but they're always expressed in 20% of the cells. Like,
yeah, I mean, it removes sparsity. I mean, it removes sparsity insofar as we can detect that by what it's doing on this projection to the basis of eigenvectors.
But then maybe I can claim that some of the sparsity is actually real. Like,
yeah.
Biologically relevant.
I mean, yes. That's right. So it could be the case that some of the sparsity is biologically relevant. It could, you also have, I mean, a related issue with this kind of procedure in general is that, I drew these pictures about, the motion of the eigenvalues as a positive thing. But of course, what that means is this is very sensitive. And if you have outlying noise, you also are going to pick that up. And so, so yes, I think that's an issue.
In some sense, there's an unidentifiability problem, right, Artosh? Like, it could be sparse because it's genuinely only expressed in one cell type that's not well represented, or because of some technical issue capturing this kind of RNA molecule with this technology. And if we don't know how to tell the difference, maybe we're better off not trying and just considering this too hazy to use in our choice of principal directions. Maybe.
Yeah, I guess my point was very naively that if you know, if a sparsely expressed gene can be predicted somehow, then maybe we want to keep that. But if it's sparse and it's completely uninformative, then it's okay to throw it away.
I mean, that's yeah.
I don't have to formalize that.
Yeah.
Do you imagine using this as a sort of pre-processing step where you go down to an intermediate dimension as opposed to sort of trying to interpret the result?
So I think
more of a
that,
denoising?
that's right. So I think the most long term. I think the most. I mean, so if you look at, if I chose not to use a projector because I don't. I talk too fast if I do that, but also I don't think it's necessarily informative. I could put up some pretty TSNE colored pictures of what stuff you come out of this, but who learns from that? But so I think that that's exactly right. That the real application of this kind of stuff is a preliminary dimensionality reduction step where you throw out a bunch of stuff that you are almost sure is noise and don't worry about exactly where you're picking the threshold, and then do something else that's fancier. Or
Project away the smallest I can.
exactly. That's right. And where you're not making sharp choices about where the cutoff is, but you're way into the bulk and you know that stuff is garbage and you toss it. I suspect that's probably the.
That seems like a practical way to leverage these observations.
So, yeah.
So, when you apply this to real single-cell data, do you actually see that the sort of large eigenvalues are actually localized?
Yeah.
Have you looked at what genes kind of develop? Is it
I mean,
biologically informative, or is it?
It appears to be biologically informative in the sense that it does very well on identifying biologically meaningful cell subtypes. For instance, in blood data, and the tests in the paper that I'm really talking about, we used blood data and we used neural data, but neuronal data. But I think that there's a team of people working on using this in bladder cancer, where again they're finding subtypes of bladder cancer that are clinically significant that pop out of this. So yeah, I mean it's seeing something. Now, as always with these sorts of things, I don't think that you can say, is this the only way you could have seen that stuff? I don't, who knows? But it is seeing something.
I think this localization phenomenon, right? You're describing it in terms of the directions we're learning in single cell. The same kinds of ideas come up in human genetics, looking at principal components. Is this component localizing on some part of the genome, or is it really just a broad ancestry thing? That's kind of second-order information maybe we don't always recognize as interesting signal we can go after the primary analysis. That's going to potentially point at some real biology.
In genetics, it's a little different. Like a signal of drift, which is actual population structure, is also totally delocalized.
Right. But not all principal components of population data are that way.
Right.
The ones that aren't.
There's the artifacts, the noise, and then there's also sort of when you have admixed samples, you get them concentrating on haplotypes.
Yep.
You have no noise model, so really, I don't know if we're saying that you can get rid of the noise, or you can get rid of just anything that if it is signal, then it's too small to find anyway.
Well, the noise model in some sense is coming from the assumption that the assumption is that what you're looking at is a low rank signal and a large random matrix, and that random matrix was sampled from random, and that's your noise model, which is sampled random entries.
Below the low rank
That's right.
subspace.
That's right. Is noise.
Not findable.
That's right.
For example, from the actual counts being Poisson with some true
Exactly.
thing,
Any other questions?
Yeah.
I was wondering if the biological interpretation really changes substantially when you compare this method with like a common practice that you use in single cell, which is fixing the principal components to just like 30 or 50, for example.
Yeah, I mean, it does better on these labeling tasks.
I think this is one of the most important things.
Yeah, I mean, so, on the tests we ran, and again, you can see the specific details of this, or I can point you at the specific details of this. On the tests we ran on these labeled data sets, it does better than other things. Now, does it do, as a personal story, I grew up in the AI lab in the old days at MIT, before I here at NE43, before the computational linguistics revolution had happened. And where people were trying to implement Chomsky's X-bar grammars. And it was very jarring to see the sudden shift to where computational linguistics is about, oh, I have this, if I add this tweak to my probabilistic context-free grammar, I get 93% rather than 91% recall on some library of tasks. And so, so I worry that, of course, the kinds of comparisons that we do to justify this are more like that than more like real qualitative improvements. But they're big, actually. You really do a lot better.
Yeah, I think what you're bringing up now it points to something that our community recognizes as like a critical task, which is to think about how we could reasonably understand and benchmark the variety of methods. I mean, there's a huge zoo of ideas coming out, right, around how we could model single-cell data. It's a new kind of data, and it's really exciting what we can get out of it. Like learn hundreds of years of morphology biology from one sequencing experiment. So. What would you like to exist as a way to compare this to other methods? Like, and as a mathematician, like, how would you want to approach the problem that you just brought up, which is you can test one other thing in your time you have for your paper, but you can't try all the things. So what could we create as a community?
I mean, so I should say, yeah. So right. So I don't know the biology all that well. And for me, the kind of thing that would be most important is, and I think I think my co-authors, Raul in particular, know the biology very well, but so he might have a different answer to this. But what kind of thing that I would say was that it would be nice to understand what a qualitative improvement looks like. Right? And have data sets where what it means, just to say, what does it mean when you really did something better? And this is hard, right? The machine learning community struggles with this, right? And the test data sets are often not good. You end up with this thing where people are optimizing for these small numerical improvements on benchmarks, which I think is counterproductive. But so having some sense of data sets where different methods are really going to have qualitative differences in understanding what comes out, I think would be helpful. But of course, that's
Yeah, and it's fundamentally different in most of those predictive tasks, because it's not like humans don't do a good job of labeling a cat or a dog. Here we really don't know what the cell types are. We just don't know. We're trying to learn that. So it's unsupervised, and that makes it a much harder question. So something just maybe to leave the audience with is, we have Kaggle, which is an amazing framework, so people all over the world can compete on these predictive tasks, and it's very easy to measure who's doing better. You just have a test set, a training set, whatever. What does unsupervised Kaggle look like? How do we build that? It certainly has to involve like a much broader community than just one person writing an algorithm because of this exact question. How do we even tell if we're doing any better?
Right.
And I don't know the answer. I have some ideas, but I think that's something we need to think about.
I think I have an answer, but it's probably not the answer to your question. I think we need to have the equivalent of Sanger sequencing for single cell. We need to have something which is very expensive, very slow, but extremely accurate.
So
I think that's the only way to do it.
For sequencing, at least, there's an answer. That we
For
know.
sequencing, there
There's
is.
that's a physicist answer,
Yeah.
Prakash. Thank you. Yeah, that was like, we just need to build a particle accelerator, and then we'll know, and it'll be good. But, and while we could build a particle accelerator, and we should, and we do, we should probably also think about simulation. Different kinds of simulation that biologists will yell at us about. This is not realistic in this context or that context. But we have some ground truth. And then there should probably be something in between that's not your platinum standard.
Yeah.
But a battery of things where we have pretty good labels, and we could ask for this whole litany of methods. When you put it out and publish it, it should be run on every single one, and we should see exactly how it compares to everything else that's ever come before. And then we do gradient descent on methods. Right?
Look, for molecular biology, we just don't know what ground truth, what the form takes, right? For sequencing, you know what form ground truth takes, is Prakash's point. But I would say for these molecular directions, you just don't even know what form.
That sound fun?
Yeah.
Something would be fun if it existed. Right. No, I think this, and tying this back to the earlier talk, this is also, this is an issue with any of these also with these geometric inference things, which is that you don't know what kind of answer you're looking for, what kind of structure, you see this structure and you don't know what it's supposed to be, because you don't, I mean, I give this example of phylogenetic trees, which is easy to understand, but in general, you have, for this kind of data, you have no idea. I mean, and if you see this geometry, I mean, the best way, what can you do? Some poor graduate student maybe has to do experiments to see if it's clinically significant, but it's hard to know what you're supposed to do.
So people have to talk to each other, right? People with different sets of skills.
Yeah,
Andrew,
right.
thank you so much for your talk.